Hi everyone. I'm just going to talk to you briefly about our road to Damascus, if you like. Our aim to try and understand SUMO, a small ubiquitin-like modifier protein, and its role in adaptive responses in plants. So, trying to give you a snapshot of our journey, and also about the kind of strategy we took to try to understand this system. So, our original aim has always been about trying to understand how plants adapt to the environment. Here's a tree. You can see in the Lake District, Lake Windermere in the background, and you can see on the right-hand side, there's branches coming out. On the left-hand side, there are no branches at all. This kind of captured my imagination, try to understand, the wind's blowing in one direction, it actually changes because plants can't run and hide, changes how the architecture of the plant and its growth and its adaptation to the environment is really finely tuned with its environment, with conditions in the environment. So to try to understand this, obviously working on trees is going to take way too long. So I decided to use Arabidopsis instead. So you can see from the screen there that you could actually control the growth of Arabidopsis really quite easily with an amount of salt on an agar plate. And as you increase the amount of salt from a low amount of salt to a high amount of salt, you can actually not only change its growth rate, but also its architecture. So this has kind of inspired us to try to use genetics to understand how we can come up with understand mechanisms involved in this growth control. So we then did what do you call an activation tag screen, whereby where you randomly activate genes with enhancer elements randomly inserted across the genome, and they activate genes within the context of the gene expression itself. So in other words, it's not ectopic expression, but is expressed higher, much higher than it would within the system that it's expressed in. So therefore, on the left there, you can see, I don't know if you can see my screen. On the left, you can see Arabidopsis seedlings do not germinate at all at 80 millimolar salt, but the mutant here that's overexpressing a specific ubiquitin-like protease is able to overcome this suppression of germination by salt and grow quite efficiently. So we then started to understand what this ubiquitin-like protease is, and this is where our kind of journey starts, really, on trying to understand SUMO. So this specific ubiquitin-like protease belongs to a class of genes, proteins which modify ubiquitin-like protein. So we then started to understand what ubiquitin is. So here's a structural information here provided, gives you the main structure of these small proteins which modify the proteins. And the archetypical one is ubiquitin here. And it's called what we call this ubiquitin fold, where you have these 3 beta sheets with the alpha helix going across it. And you can see this fold across this ubiquitin-like proteins like RUB, related ubiquitin, hub, and here you see SUMO, which is small ubiquitin-like modifier protein here. And the main differences is that it's got very long N-terminal extension, and then it pretty much a ubiquitin fold, as you see here, and a diglycine motif here. And unlike ubiquitin, where there are about 11 ubiquitins made in tandem copies, where ubiquitin or SUMO is made as a single premature SUMO, which is then processed to give you mature SUMO. I think one of my postdocs, Anjul, has spoken about this quite in one of the Garnet meetings. So I'm just going to probably go over it quite quickly here. So there are 8 SUMOs in plants, and SUMO 1 and 2 are the major SUMOs which are then processed to get conjugated to target protein. And I'll show you in a minute what that means. But then there are other SUMOs, SUMO 3, 4, 5, 6, 7, and 8, which do very different things, which I don't have time to go into now. I'll just focus on what SUMO 1 and 2 are doing to environmental adaptation in plants. And SUMO 3 has also a role and has been shown to conjugate to target proteins, but their real function in adaptation is unclear at this stage. So you can see SUMO 1 to 7, SUMO 1 to 8 are pretty much quite well conserved at the C-terminal end, but not very much at the N-terminal end. This particularly at the C-terminal end, it becomes even more important for SUMO 5, 6, 7, and 8, where there are really great variation among the SUMOs. So that's a separate story that's emerging out of that now from different labs. So hopefully next time when we're in this Ghana talk, I might be able to tell you more about what SUMOs 5, 6, 7, and 8 are doing, what SUMO 4 to 8 are doing. So how does SUMO get conjugated to target proteins? By a series of enzymes, really. And this series of enzymes starts with SUMO being made as premature SUMO, like a precursor SUMO, which then cleaved at the C-term to expose the diglycine motif here. And this is cleavage is done by a set of proteins, SUMO proteases, which the OTS1 belongs to, is the ubiquitin-like proteases. And these then expose the diglycine motif, which is then taken over by the SUMO E1 and the E2 and then the E3s, which then conjugate SUMO onto lysine residues on target proteins. And cysteines are also can be conjugated to SUMO, but somehow the cysteine-glycine link is less stable. Okay, and then you have the pile domain proteins here, which act as E4s. And these E4s extend this chain, SUMO chain, on target proteins. And that's a class of SUMO proteins, which I'll go into a lot more detail in a minute, which cleave only SUMOylated, SUMO from the target proteins, but do not take part in SUMO processing, while the ubiquitin-like proteases can do both release, cleave SUMO of the target proteins, but also process SUMO. So these are new classes of SUMO proteins that we've identified, and we'll go into a little bit more detail on that. And then you have the stubble proteins, which are ubiquitin E3 ligases, which do not form the main part of the SUMO pathway, but are important nonetheless, where what they do is that they target SUMO conjugated proteins, target proteins for ubiquitin mediated degradation. So they are ubiquitin E3 ligases, which specifically target SUMOylated proteins for degradation. Now, the key thing to note about this compared to the ubiquitin system is immediately becomes apparent is that this very small number of ubiquitin E3, SUMO E3 ligases. So far, there are 2, and perhaps if you count the pile domain proteins, the E4s, there are 4 SUMO E3 ligases, if you like, E4 E3 ligases. Okay, in plants, in Arabidopsis, and also in crops. The types there are only 4. In rice, for example, and we're still working out how many there are in wheat, and also in maize, about 4. But then the ubiquitin E3 ligases in plants, there are closer to 2,000. So you can see the shift has gone from the gene expansion is no longer at the E3 ligases for the SUMO system. But then the SUMO proteases are about 16 so far we've identified in Arabidopsis. So I'm sure there'll be more coming up soon in the next few years, because this field is just emerging and its importance is just beginning to be understood now. But you can see from the organization of the gene clusters, the gene system, that the protease gene family is expanded, while the E3 ligases, which conjugate SUMO on target proteins, has remained the same, while the proteases which process SUMO as well as cleave SUMO off target proteins gene family has expanded. So we think that the SUMO proteases at this stage provide the real clear route for specificity within the SUMO system. And there are 2 types of proteases, the SENP type, which is the ubiquitin-like protease, the ULP, and then the DESI type. So I'm going to give you a couple of examples of the ULP type and DESI type and how they really very specific, have a very specific role in different aspects, even in one particular pathway. Okay, so they're very different catalytic domains and they do very different things. Okay, so the OTS1 belongs to the ULP type SUMO proteases, and there are 2 of them in Arabidopsis. When you knock them out, you get a very sensitive plant growth, sensitive to salt plant that grows very slowly, growth retarded. And what this slide is trying to tell you is that we've made very SUMO1 to specific antibodies, and we can run them on a gel and immunoblot and do immunoblots of those antibodies to look at SUMO conjugation. This smear up here that you see at the top right-hand corner, I should use my laser pointer really. You can see here that the SUMO conjugation pattern is really down to the SUMO proteases that accumulates in high levels. And what plants try to do is a Western blot here that's showing the tag form of the OTS SUMO protease, that the plants, when they see stress, in this case, salt stress, they want to get rid of the protease. So the system in Arabidopsis, the way it's organized, is that in specific stresses, particularly in terms of salt or drought or pathogen or UV light or different kind of stresses, tend to degrade very specific protease. And that's what we're beginning to learn now. And that then gives rise to very specific set of SUMO conjugated proteins accumulating, and thereby then modifying the response of the plant to that particular stress or adaptive environmental cue. I think that's what we're beginning to learn here. So, and that can be summarized in this kind of slide where we can show, for example, when there's normal, when there's not, when you don't experience stress, and that's not really true, plants, because they can't run and hide, you're always seeing some kind of stress in a plant. Well, ambient environment, if you like, and then when the stress environment, they slow down growth, and you see this accumulation of SUMO conjugated target proteins, and then that affects how plants grow, and you can control that SUMO conjugation by controlling the particular stress that you can have, and you can find out what those targets by mass spectrometry, and then identify which lysine the SUMO are conjugated in, and then start learning about how SUMO then controls that particular or impacts the particular adaptive response, if you like, in this case. And what plants tend to do is to get to this SUMO conjugated state by primarily getting rid of the SUMO protease. So that's the signature that we're starting to see in the different stresses. And in this case, we use growth as a very nice example to identify SUMO conjugated proteins which accumulate when that protease OTS1 disappears, which then blocks growth under salt stress. And using that system, by understanding, in this case, I've shown you only for the OTS1 SUMO protease system, that we've shown, for example, different kind of SUMO protease, in this case OTS1, has very specific role in, for example, root patterning. Unfortunately, that video is not running, which I can't seem to understand why it's not running. Hydropatterning in roots, brassinosteroid signaling in plants, as well as dwarfing through the GA pathway and also JA pathway, and Leyden light signaling. So, as well as also defense responses in plant, which I'm going to talk about for, in the next few minutes. So one specific protease has really very specific roles in those different adaptive responses. So I'm going to talk about this one particular one here in the immune response in this set. So we were starting to understand the role of SUMO proteases in the immune response. So what we started to do is to look for SUMO proteases which are impaired in the PAM-triggered immunity, in this case through flagellin. And flagellin binds to, it's the bacterial ligand from the bacterial tail, which then flagella, which then binds to FLS2, the pattern recognition receptor, which then binds to the co-receptor BAC1, which then allows signaling to occur by release of the BIC1 kinase, then the oxidative burst, and then the BIC1 kinase then switches on a set of MAP kinase. This immune signaling switches on MAP kinase cascade, which then gives rise to changes in transcriptional reprogramming to give you overall the PAM-triggered immunity response. And this kind of simple model that tells you, gives us a framework to see where SUMO might sit in this pathway. So what we found was one of the targets when flagellin binds to FLS2 is FLS2 itself. So FLS2 is conjugated to SUMO1 within 15 minutes. And this conjugation is really is really important for immune responses. When you mutate the SUMO side where SUMO is conjugated to FLS2, the oxidative burst is abolished in the bottom here. And the plants are very compromised for PTI. In other words, PSD DC3000 is even more virulent in FLS2KR plants, which no longer can conjugate to SUMO, while the wild-type plants is able to complement that phenotype. And what, long story short, we published this a couple of years ago, is that the SUMO conjugation onto FLS2 allows BIC1 kinase to be released from the pathway, from the FLS2 BAC1 kinase. And this release then allows the oxidative burst to happen and immune signaling downstream to occur. And if you don't have FLS2KR here, where you cannot release, or if you don't SUMOylate FLS2 in a FLASH2 dependent manner, the BIC1 cannot be released from FLS2. And therefore, we have now provided a step whereby SUMO conjugation onto FLS2 is required for the BIC1 to be released, BIC1 kinase to be released from the BAC1 FLS2 co-immune receptor. So that's where we're at with that. And what we found was that there was this really specific SUMO protease, Desi3A, which is an isopeptidase type of SUMO protease. In other words, those that does not process SUMO, but it's very different type of SUMO protease, which only cleaves SUMO chains or SUMO conjugated to target proteins. And this is the Desi type of SUMO protease, which have very specific catalytic domains, which allows us to distinguish them from the SENP type or the ULP type of SUMO proteases. And in order to prove that this Desi protease is involved in the immune process, is that we've shown we made SUMO chains in vitro, we purified the SUMO1 chains, and then we incubate with recombinant Desi3A. And that then cleaves the SUMO chains from here and makes the monosumo from this. And this is a middle plot showing this very nicely. And we mutate, this is the cysteine protease like other ULP SUMO proteases, and we mutate the cysteine to a serine, you no longer works as a Desi SUMO protease. And this obviously, this Desi ULP SUMO proteases do not work on SUMO chains, and Desi SUMO proteases not necessarily work on a monosumolated target. So they have to be SUMO chain in this case to be working. So we suspect FLS2 is SUMOylated in a chain manner that allows Desi3A to work, and that becomes a different story altogether. So we've shown that the Desi SUMO protease with M-cherry tagged in this stage co-localizes to the membranes like FLS2. That's what this slide is showing. And when you mutate the Desi3A, when you CRISPR it, plants are hyper potentiated for immune responses because they are hypersumolated for FLS2. And the MAP kinase cascade is activated more in this case compared to FLS2 or the co-0 background. And the plants are more sensitive to flagellin on plates, but the root growth is inhibited, unlike FLS2, which is more sensitive to flagellin on plates. And when you cross it into the FLS2 mutant, so when you put them on flagellin, it reverts the phenotype. So the Desi3A flagellin growth sensitivity on flagellin plates is working through the FLS2 receptor. And that's just quantification. The oxidative burst is also more potentiated in the Desi3A background when you treat Arabidopsis leaf disc with flagellin. And that again goes through the FLS2 pattern recognition receptor. So what we found very specifically that like I said before, FLS2 is hypersumolated. Again, this is a SUMO blot showing that there's a lot more simulation here in the Desi3A background for FLS2 than in the wild type background, in the FLS2 mutant background, complemented with the FLS2 on its own. Okay, and what was intriguing, was very clear for us, is that when you treat plants with flagellin, like when you treat salt, you see OTS1 disappearing, protease disappearing. We see here the Desi3A disappearing within minutes. And we think that coincides with FLS2 being hypersumolated because the protease that cleaves SUMO of FLS2 is no longer there. So FLS2 is hypersumolated, and that allows facilitates the release of BIC1 kinase from the BAC1 big FLS2 co-receptor complex, and that then allows downstream signaling to go on. Now, that's where we're at the stage where we're allowed to put SUMO on this stage here. And what we try to understand here is that for a very small number of genes, 34 genes, SUMO is able to like really be involved in quite critical stages of the immune response in this case. And that's also true for the other adaptive responses that we're beginning to understand. And what we tried to see is that does SUMO also play a role downstream to affect transcriptional reprogramming? If so, is there a SUMO encoded pathway within the pattern PTI that we may be able to unearth? And not only that, can we then use the SUMO system to understand the pinch points within PTI or even ETI in this case? And that's the aim going forward for the lab. Now, so what I'm going to talk to you about is this new types of SUMO protein, another type of SUMO protease called SPF 1 and 2. And these are ULP type, again, going back to similar to OTS type of SUMO proteases. And what we found when screening for pathogen response, we found that the SPF 1-2 double mutants are very resistant to botrytis. Remember, BIC1 stands for botrytis-induced kinase 1. So we started to screen for botrytis as well for in our SUMO protease mutants, and we found these SPF 1-2 double mutants. And they are very resistant to botrytis, while normally Columbia wild-type plants are very susceptible to botrytis infection. And the WORK-E33, which is one of the key transcription factors which are required for BIC1 response, not BIC1, botrytis response in Arabidopsis, where knockout of those is very susceptible, while SPF is more resistant. What we found was that in the SPF mutants, WORK-E33 transcripts are very highly induced. And this is also true when you do botrytis infection, but also when you treat the Arabidopsis seedling with flagellin. In the SPF 1-2 knockouts, there's a lot more transcripts of WORK-E33 compared to COL0. So we think there may be a connection. We thought there may be a connection between WORK-E33 and the SPF SUMO proteases. And of course, WORK-E33, long story short, is also induced by flagellin, the protein, and it's induced within minutes, similar to what we see in the SPF mutants, where there's a lot more WORK-E33 transcripts. And we've shown that WORK-E33 as a transcription factor that interact with E2 enzyme, SUMO E2s. That's what this immunoblot is showing. WORK-E33 comes down really nicely on transaminase in Bent with the the SCE, which is SUMO conjugating enzyme 1 or the E2. And it interacts with SPF 1-2 SUMO proteases. And that's what this immunoblot is showing here. These two blots here, that they come down very nicely with GFP tagged WORK-E33. What we found is that WORK-E33, you can purify the E1, E2, E3s, and you can incubate with the recombinant WORK-E33, and it very nicely SUMOylates WORK-E33. And we can identify the SUMO sites of WORK-E33. And before that, we even can show that when you infect with botrytis scenario, in the case of ours, we see WORK-E33 SUMOylation going up here. This is WORK-E33 protein, GFP tag, and then it's SUMO conjugated WORK-E33 going up here. And we see very similar, but a very different pattern, a similar SUMO conjugation, but very different pattern when you treat Arabidopsis seedlings, flagellin 22. And you can see this smearing of SUMO conjugated WORK-E33, which you don't see with botrytis scenarios. So it's a different kind of pattern. And that's a different story that we're trying to chase up now, where we think there are effectors going in botrytis targeting WORK-E33 for cleaving the SUMO off the target proteins, right? SUMO off the WORK-E33 target protein. And you can see here within minutes, we can start to see SUMO conjugated WORK-E33 accumulating. And we can find the SUMO sites and we can mutate them. And the SUMOylation here, it's gone. This is WORK-E33 3KR. There are 3 SUMO sites spanning across the protein. And when you can mutate them, the SUMO conjugation is no longer there. And we can also show that we can immunopurify these proteins and incubate them, SUMO conjugated WORK-E33, incubate them with the SUMO protease. And we can see here that they start disappearing. WORK-E33 actually interacts with SUMOylated WORK-E33 and cleaves the SUMO off the target WORK-E33. And when you make WORK-E33 non-stimulated form, put them back into the SUMO WORK-E33 knockout plants, and they become a lot more susceptible, like WORK-E33 knockout themselves. So it's almost as if you don't simulate, WORK-E33 is no longer effective in performing its transcription factor response effect to botrytis cinerea, as well as the flagellin to switch on PTI signaling. And here's the problem that we had here. Once we know that WORK-E33 non-SUMO form is no longer functional, we found that the protein was stable between the non-stimulated form and the stimulated form, and it was stable, but it was not able to perform its transcriptional activation role. So it was able to bind, and that's what these slides are doing. The protein is stable, 3Kr to wild type, but its ability to switch on WORK-E33 itself. So the WORK-E33 transcription factor sits on the WORK-E33 promoter and switches on WORK-E33 itself. And that the simulated form can't do. And that's what these chip assays are doing to show the induction of WORK-E33 in the WORK-E33 by the 3Kr is very non-simulated form is very much hampered. And what it does is that what we've shown here is just summarizing what I've said. It doesn't affect localization, doesn't affect stability, but doesn't affect its DNA binding as well. So we can find, for example, SUMO WORK-E33 is really the ability to bind to the promoter itself is not significantly affected the SUMO form or the non-SUMO form. So we can, the PAD3 promoter enrichment of the transcript factors doesn't greatly affect it. But its ability to switch on PAD33 is greatly affected. In other words, we know that the WORK-E33 transcription activation ability, transactivation activity is gone. And that is normally, activity of WORK-E33 is normally conferred by phosphorylation by the MAP kinases. And that's what's been shown by several different labs. So we think SUMO has a role of bridging the MAP kinase cascade, MPKs in this case, to interact with WORK-E33 and then to activate its transactivation role. And that is mediated, what we found is mediated by simulated WORK-E33 interacting with MPK3 through the SUMO interacting motif in MPK3. And that's what this slide is showing here. So it's a new dimension of signaling whereby you get WORK-E33 simulated proteins are able to then selectively interact with MPKs or different proteins which have a SUMO interacting motif. So SUMO conjugation or SUMOylation is a covalent interaction. So this is an isopeptide bond, and SUMO interaction with SUMO and SUMO interacting motif is a hydrogen bonding. So that's a non-covalent interaction, a very flexible interaction. And we model that in the MPK3 here with SUMO1 to identify very specific the SUMO interacting motif within the MPKs. Identified it, and we mutate the isoleucine, and that's what this slide is showing here. This is a SIM motif here, and we can mutate this really very specific isoleucine here. And then we can show that SUMO binds to MPK3 in vitro. This is a Western blot showing that with his tag SUMO1 and GSD tag MPK3. And then when you put in the SIM mutant, this interaction greatly is reduced. So MPKs contain, in this case, 3, 6, and 12, contain a very clear SIM motif that binds to SUMO, and you can mutate it. And we've done the same here, whereby we put the MPK SIM here. In vivo assay, we pull down the assays here. All I'd like you to focus on is the WORK-E33 here, GFP tagged, and the HA tag MPK3. They bind here clearly on when you incubate both of them together. And then when you have the SIM motif, this interaction WORK-E33 is gone greatly with MPK3. Okay, but another transcription factor, which is a target for MPK3, which is not involved in immunity, but it's more to do with speechless in this case, more to do with development or patterning on giving rise to changing pavement cells on the surface of leaves to guard cells, has no role in the interaction of MPK, the SIM motif. So SUMO in this case, during the immune response, has no role with its ability to interact with speechless. Therefore, what we've done here is what we found is that during the immune response, SUMOylation of specific targets like WORKY33 is able to, MPK is able to selectively pick them out through the SIM motif, through its SIM motif to directly bind the simulated proteins. And that binding allows enhanced phosphorylation of WORKY33. That's what this slide is showing you. This is a phosphatic gel, while the SUMO33 non-simulated form that is not able to bind to MPK3 is not hyperphosphorylated. And this phosphorylation linked to SUMOylation then activates WORKY33 transactivation activity and switches on PAD3 transcription factor and switches on PAD3 transcription. So what we've shown here is a new kind of way of looking at signaling, where SUMOylation controlled by SPF1 to SUMO proteases on WORKY33 is then nullified when you put flagellin signaling, enhances WORKY33 SUMOylation, which then allows it to then interact with the MPK signal cascade and to allow phosphorylation to occur, and thereby then switch on its transactivation activity. So we then may have a SUMO encoded pathway here, whereby you have a type of DESI SUMO protease, which then, upon flagellin binding to FLS2, starts the signaling off right at the membrane SUMO through a specific set of DESI type SUMO proteases, which then all the way through down here into the nucleus, whereby SUMOylation of WORKY33 then gives rise to transcriptional reprogramming. And we think there are pathways within this pathway from the membrane all the way to the nucleus where there is a SUMO coding going on. And therefore that gave rise to the idea whereby our new grant, where we're looking at, for example, that using Arabidopsis roots for the system, where we are generating, if you like, where the environmental signal is perceived by SUMO. So in this case, pattern recognition receptor like FLS2 may perceive environmental stress, and they're coded by DESI SUMO proteases regulating SUMOylation of FLS2, and then going to the nucleus. And then at some stage, similar thing occurs in the nucleus where WORKY33 is SUMOylated. Where the perception occurs and how it's detected by SUMO, and how then it then reprograms SUMOylome within the cells to find specific hub proteins that are SUMOylated, which then gives rise to specific phenotypes in the plant, then to enact the adaptive responses. And this is our new grant that we're going to start working on this year. And this is in collaboration with Nottingham, Liverpool, and Cambridge, where we're going to generate the first SUMO cell atlas in plants, where we'll map out the entire SUMOylome and the entire SUMO machinery and how it responds to different kinds of stresses in the Arabidopsis root. And we'll make all of this publicly available, and then develop a programs whereby we should be able to tell you which targets are SUMOylated under which conditioning, which cell types, which then gives rise by, we're going to try and do a whole bunch of single cell SUMOylome and single cell transcriptome to see which targets are SUMOylated in which cell types, which give rise to specific adaptive responses. And I think that's the aim of this, this grant. There are postdoc positions available. If you're interested, please email me. And we also have a website, SUMO Code. If you search for us, you'll find us there, and we'll start updating the SUMO Cell Atlas. We've just started last month, and watch this space for the SUMO Cell Atlas. And I think, and the PIs of the grant and the co-Is are all listed there, as well as the SUMO Code website. Please visit us so you can learn more about the project. And we'll be starting to generate tools that will be very easily make it available to the entire Arabidopsis and the rice community to be able to use this system. Last thing I want to say here is just want to thank all the people who did the work, the people in the lab. A lot of the work I talked about today was done by Momita, Angel, Beatrice, and Vivek. These are the main postdocs, but ably assisted by a whole bunch of people here, that too many of them that are to name here really. But a special mention to Chunjin and Alberto and Momita for working on those projects and drive them through, and my collaborators, Malcolm, Patrick Krysen on the MPK pathway, Malcolm Bennett on roots, and Lucio, who started off the SUMO thing with me. I started my group in Glasgow, and all the collaborators who helped me out in the future, and those funding bodies like ERC and UKRI who funded my work over the last few years. Thank you very much for listening, and I'm happy to take any questions. I think I'll stop sharing.
No, you can keep, well, it's up to you. Keep it on if you wish. That's fantastic talk. Thanks very much. It's really great to see, how far sumo has come in the last 15 years, really, when I think we started working on it or started looking at it, it was a bit of a novelty and wondered what was going on, but obviously very important. So let me start off, ask a question for you about these sim domains. So, it's a 6 amino acid motif, and there's going to be a lot of those motifs throughout the proteome. So do you know how many of these motifs are bound by SUMO, or is that something you're looking into now?
Yeah, this is one of the things that we're looking at the ground. At the moment, we have a software program that allows us to faithfully predict SUMO interacting motifs in any plant protein, and it can be extended to yeast as well. Mm-hmm. And we are starting to see similar things happening in animal proteins. So we'll be able to predict. The core is about 12 amino acids, and normally sits on a beta sheet, and it's a hydrophobic core. So if you disrupt the hydrophobic, it's a very unusual structure of a hydrophobic domain, hydrophobic beta sheet sitting on the outside of a protein. So structure information can be useful, but we have software that allows us to predict that at the moment. And we are finding lots and lots of these peptide, these motifs across different proteins in plants. And we'll make all of these available as the program commences.
Okay, so I encourage people to ask questions in the question box. We've got a couple have come in already. So first question from Bemi, and they ask whether SUMOylation of FLS2 mediates the FLS2 BIC1 interaction. How about BIC1 phosphorylation levels in the DESI3A mutant in response to flagellin 22?
Yeah, I mean, this is things that we are testing at the moment. The BIC1 phosphorylation is a good thing to look at. And also the key thing that we see is that the oxidative burst is completely abolished in the FLS2KR plants. So we think there is a link of not only SUMO releasing BIC1 from the FLS2 back on immune complex, but also affecting the BIC1 activity in terms of switching on this oxidative burst. So that's what we're looking at this stage. And that's a very, very good question. And hopefully in the not so distant future, we'll be able to say something about that.
Okay. All right, question from Sam Mugford, and he asks, he says, great talk, Ari, I agree. Do you know how SUMOylation of FLS2 interacts with the endocytosis or degradation pathways?
Yeah, so the FLS2 KR plants, the FLS2 is really, it's very hard without doing all the controls and repeating many times. We are beginning to see that it does recycle much quicker than the wild type itself. Mm-hmm. So what I'm trying to say is that it's less stable. FLS2 KR non-SUMO form, it's less stable. Whether it's going down the recycling or the endocytosis route, I don't know. I mean, we are open for collaboration. We're not looking down that route, and it'd be great if we could collaborate with someone to understand that pathway.
Cool. So a bit more of a general question from Wendy Moore. She asks if this pathway, so the SUMOylation pathway, is active during embryogenesis. Do you know?
Yes, because some of our SUMO knockouts, protease knockout show, well, one of the targets is Monopterus as well of SUMO. So Monopterus has got an embryo phenotype, I think. I'm not wrong, I could be wrong, I apologize for that. And we do see auxin targets as well. So there is, we know, for example, if you knock out the E2, the plants are embryo lethal. So it has a role in the embryo development.
So can you generalize about the fact that there's so few SUMO E3s and so many ubiquitin E3s, and what's the differences between those systems?
So SUMO's just decided to go down a different route. So basically, what we think is happening is that's what the program aims to understand, is that we get molecular hardwiring. As soon as you see stress, SUMO is then sprayed, if you like. I use the word sprayed very casually here, apologize for that, sprayed because we don't understand how that occurs across the whole bunch of proteins. And then the plant, depending on the secondary signals that comes along, the plant then decides which are the targets to take the SUMO off to enact a particular pathway signaling. And that's where the proteases come into play. And obviously, there's not enough proteases in itself. We've got 16 so far, right? So there's going to be overlaps of within the proteases, functional overlap between them. But what we're seeing is that there is functional overlap, but not a lot. So there are specific pathways that are up that are affected by specific proteases. So we think that this molecular hardwiring or spraying with SUMO and then taking SUMO off specific targets, and these targets are then going to be hubs, depending on what pathway you're looking at, is the way to understand how SUMO encodes signaling in the plants. I think that's the way to go forward. It's connecting the protease to the SUMO conjugation at the cellular level. And that's what the program aims to do.
That's a lovely image, that the spray, you got quick, off it goes, and it goes everywhere. So just a couple general kind of questions to finish from William Rooney. So first of all, he asks which came first, SUMO or ubiquitin, and then secondly, is SUMO only in humans, plants, and yeast, or is it in other organisms as well?
Yeah, you see SUMO in all eukaryotes.
Okay.
And SUMO variants in prokaryotes as well. In all karyotes, you see them SUMO. Ubiquitin definitely came first, because if you go back further back, you start to see things that look like ubiquitin and SUMO, and they look more like ubiquitin than SUMO. We think ubiquitin came first. That's clearly established, I think. I just don't have the knowledge at the moment in my mind to tell you where they branched off. And then SUMO is then adapted from ubiquitin. And the SUMO machinery is also adapted from ubiquitin pathway. And SUMO proteases do not cleave ubiquitin. They do not act as ubiquitin proteases. Right, so it's a different system altogether.
Cool. Okay, that's great. So I think we'll leave it there. Thanks very much, Ari. I'm sure